my dears little champions, I hope that you are doing wonderful as usual. So we thought we would be also doing something for children who have opted for biology in KCET, and that's why we have come up with this thing, that is solving top 30 questions for the chapters, for the most important chapters. And the first chapter that I took is molecular basis of inheritance. I was saying molecular basis of importance, so you see how much importance it has, right? So basically, yes, so today we would be solving 30 questions from this particular chapter. You can try solving out by yourself also. When I am trying to show, you can pause the video, try it out, and then check your answers with me, my dear children. So let's get started, and if you think that this particular thing is going to be very helpful for you, do not forget to comment it down. We will keep on going with this particular series. Yeah, now okay. So the first question, the Lac-Opron model was elucidated by, who found it, who discovered it? Jacob and Crick, Watson and Crick, Francois, Jacob and Jacqueminoid, Hershey and Chase. So this is a direct question. There is no elimination method coming over here. We should be knowing about the names of the scientists who were behind everything, right? So the Lac-Opron was basically elucidated by Francois, Jacob and Jacqueminoid. There is no question to that. But as a teacher, I should be also telling you the names of the other scientists and what did they actually do and why I chose them as the answer. So basically, Watson and Crick were the people who were behind saying that DNA has a double helical structure. So DNA helix, okay, or DNA double helical structure was put forward by Watson and Crick, and they also said one more thing. Whenever this particular DNA strand replicates, okay, so suppose this is replicating, for that it has to separate. This is one strand, this is another strand, okay. Now there will be a new strand being formed around it. Similarly, there will be a new strand being formed around it. If you see, this particular strand is from the parent DNA, this particular strand is also from the parent DNA, this is the new strand, this is a new strand. So can I say it is semi-conservative mode of replication where it is conserving the parental DNA as well. It also has a parental DNA. It's not exactly taking the parental DNA for replication. One new strand is there, so I call it semi-conservative mode of replication. Now this was also put down by Watson and Crick only. Okay, semi-conservative mode of replication. So with this, now if some other question comes that who were the people who were behind semi-conservative mode of replication, children, you know it's Watson and Crick. Who were the people who gave double helical structure, children? It's Watson and Crick. Okay, now what about Hershey and Chase? Hershey and Chase were the people, okay, who said that genetic material is definitely none other than DNA. So they were the people who finalized it by doing an experiment with bacteriophage and E. coli. Until now, there were so many experiments going on. Okay, for example, you might have heard about Griffith's experiment. Okay, he did an experiment to find out what's the genetic material, but still people were not kind of very happy. Then Avery and MacLeod came. They were not able to entirely say that this is the answer, that DNA is the genetic material. Lastly, who came, children? Hershey and Chase came, and they said that DNA is definitely the genetic material, and they did the experiment with the help of bacteriophage and E. coli. Okay, so Hershey and Chase were the people who finalized this. So I hope you know that. Now Jacob is from this, so that's why we eliminated. We are not explaining that. Crick is from this, so this option goes wrong. This is the answer that we have for the Lac-Opron. Works? Will you remember it? I was asking, will you forget it? Ma'am has gone nuts. Okay, histone proteins are positively charged, not and, are positively charged because they are rich in basic amino acid residues. First question, what do you mean by histone proteins? So basically, there are 1, 2, 3, 4 histone proteins. Similarly, at the back also there are 4. 1, 2, 3 and 4. So in total 4 plus 4, how many of them do you have? You have 8 histone proteins, or you call it histone octamer, my dear children. Octamer stands for 8. This is octamer. Okay. Now, protein, it's a protein. So protein has to be made up of amino acids. And I say that this is a basic protein. So I say that basically there should be some positively charged amino acids in there. There should be certain positively charged amino acids in there which should be making up this protein. Why do I say so? Because you know what? The use of this particular protein or this particular structure is to wrap the DNA around it. We want the DNA to wrap around it. Why do we want that? Because DNA, if it spreads like that, we would not have enough space to accommodate it inside the small nucleus that we are having. Okay, it's almost 2 meters long. We cannot have that particular space. So we need proteins which would help the DNA to coil around it. One very important fact about DNA is that DNA, children, it is negatively charged. So if the proteins are also negatively charged, will it wrap around? No, it will repel. So we need positively charged proteins. For that, this particular protein should be rich in certain amino acids. That is the question. So histone proteins are positively charged because they are rich in basic amino acid residues. What are those? What are those residues, children? It is arginine and lysine. It's a direct question again. It's arginine and lysine. You have to remember it. Arginine, lysine, Al. Okay, works. And always remember, positive and basic term always comes together. So here the amino acids are positive, they are basic. Cool. And DNA is negatively charged, it is acidic in nature. Works. So I hope you got the answer. This came in 2023. Going ahead with the next question. Eukaryotic genes are monocystronic, but they are split genes because, what do you mean? First thing, eukaryotic genes are monocystronic. What do you mean by that? Monocystronic means that basically, if I say that this is DNA, basically there will be only one gene in a transcription unit. Okay, for example, this is the DNA. And I say this is the place where transcription is going to take place. This will contain only one gene, my dear children. Excuse me, I'm so sorry. Yes. So basically, there will be only one gene that I have over here, and this gene will be coding for one particular protein. Okay, that is what you mean by monocystronic. Now, is there something opposite to it, ma'am? Yes, there is something that is known as polycystronic. What do you mean by that? Basically, when you have in a transcription unit, you have multiple genes. Okay, when you have multiple genes like this, like this, like this, if there are 1, 2, 3, 4 genes, and 4 of the genes will be coding for 1, 2, 3, 4 proteins overall, you call this as polycystronic. Okay, what do you call this as? You call this as polycystronic, and this is found in prokaryotes. This is found in prokaryotes. Now, I said that eukaryotes are monocystronic, that means there is only one gene. Then, why is it that they are split genes? So, eukaryote genes are monocystronic, but they are split genes because, what do you mean by split genes? That means the genes is in parts. Why is it so? I am saying it's one gene only, it is going to code for one protein. Then why do I say it's split gene also? Because, my dear children, what happens is that in this one gene, there will be sequences that will be coding sequences. There will be sequences which are not going to code for anything. So there are non-coding sequences as well. And there are coding sequences as well. Coding sequences as well. This particular thing will help in the formation of a protein. That one protein. This segment will help in the formation of this one protein. Whereas this middle segment which is coming, it is not going to code for anything. Okay, and what do I call it, children? I call this region as intron. What do I call it? I call this as intron or intervening sequence. And what do I call this coding thing as? I call it as exon. So in between the exon, what do you have, children? You have introns in the case of eukaryotes. So when I want to make a protein, what I do is, before I try to make a protein, I will cut this area off. I will cut this thing off such that I will be having a gene like this without the middle area, that is the intron region. And now this gene will be coding for that one particular protein. So it's all because of the intron. Whereas over here, even though they have multiple genes, they do not have introns, my dear children. They do not have introns, they just have exons. So eukaryotic genes are monocystronic, but they are split genes because introns are interrupted with mutons. Nothing. They contain exon only? No. They contain introns only? No. They contain both, right? Now exons are interrupted by introns. This is the answer. The coding sequences are interrupted by the non-coding sequences. This becomes the answer. Why introns are interrupted with mutants is not the answer, because does not make any sense. Mutants are those region which is more probable to undergo mutation. Okay, that does not make any sense. Over here we have something that makes a lot of sense. So D is the option. Works? Okay, enjoying solving this? Guys, you know what? We would be only continuing this thing if we see that there are children for biology, children are utilizing this particular opportunity very properly. So I want you to comment down that you need more of the series, or basically if you are liking it, if you're not liking it, if you want something to add more, anything that is your feedback should be there. Okay, then only probably this could be getting going. Okay, so that should be your promise. Fine. Now, fourth question. During transcription, during transcription, the DNA strand 3' to 5' polarity of the structural gene always acts as a template because. What do you mean by that? Basically, if I say this is the DNA, okay, this is the DNA, this is 3', this is a 5' end, this is a 5' end, this is the 3' end. Now I try to open it, it looks like this. This is 3', 5', and this is 5', this is 3'. Works? Now listen to me carefully. The question says, during transcription, what do you mean by transcription? When DNA makes an RNA out of it, this is known as transcription. This mRNA which is being formed help in the formation of proteins, and this is known as translation, where I am translating the information of the mRNA. No one is able to understand the language of mRNA, but there will be one guy who will be able to understand, that is the tRNA. So basically it helps in translation, and hence we get the proteins. So in a way, DNA is helping in the formation of proteins, but over here we need an intermediate guy, that is mRNA. Now during transcription, that is during this formation, that is formation of mRNA, okay, the question is that is being asked is that the DNA strand 3' to 5' polarity of the structural gene always acts as a template. Why is it acting as a template, my dear children? What do you mean by template? Template is basically that particular strand which is going to undergo transcription. So ma'am, this strand does not undergo transcription? No children, it does not undergo transcription. You call this as a coding strand. Now usually when we say that is coding strand, we get confused that this is the one that undergoes what? Transcription. But no, it does not undergo transcription. It's a template strand which is going to act like a template, and it will be helping in the transcription. If I said translation, please forgive me, okay, I meant transcription. Now the question says, why is it like this is chosen? Why this is not chosen? And the answer to that is, there is a very important enzyme that is responsible for transcription, which is known as RNA polymerase, my dear children. And RNA polymerase basically makes the RNA in 5' to 3' direction. So you know that it has to be the opposite one that should be coming over here always. So if it's saying that it is going to make from 5' to 3', then obviously it will be choosing this strand that is having 3' to 5' polarity. So RNA polymerase makes things in 5' to 3' direction, which is why my dear children, this particular strand is chosen to be the template strand. Okay, always remember RNA polymerase direction is 5' to 3'. Similarly, DNA polymerase direction is also 5' to 3' itself. Works? Now, remember one more very important thing. This RNA polymerase is making a new RNA strand with the help of the sequences which are present on this DNA. So can I call this RNA polymerase as DNA dependent? They are dependent on the DNA for making an RNA strand. How? Basically, for example, if it is A, T, G, C, in this mRNA it will be U, A, C, G. So complementary to it, it will be making a strand, which is why I say that it is DNA dependent RNA polymerase, and its polarity is from 5' to 3'. Now we have to match the option. So nucleotides of DNA strand 5' to 3' are transferred to mRNA. Is it that so? Nucleotides of 5' to 3' is it transferred to mRNA, my dear children? Nothing of that happens. Enzyme DNA dependent RNA polymerase always catalyzes the polymerization in 5' to 3' direction. Yes, enzyme DNA dependent RNA polymerase always catalyzes the polymerization as in formation, okay, polymerization in 5' to 3' direction. This is the answer. But let's read the other options as well. Enzyme DNA dependent RNA polymerase always catalyzes the polymerization in 3' to 5'. No, You saw the arrow, it is in which direction, children? 5' to 3'. So this is wrong. Now enzyme DNA dependent RNA polymerase always catalyzes the polymerization in both the direction. No, my dear children. If it happened in both the direction, this strand would also have been a template strand. So no, this is the answer. Works? So question number 4 is also solved. This came in 2023. Next question. Question number 5. Gene mapping technology was developed by. Now this is a question that is coming in Principles of Inheritance and Variation, but somehow they have kept in this particular book, in this particular column, so I thought I'll explain it here only. So gene mapping basically means where is the location of one particular gene, okay, what is the distance between one gene and the second gene, or basically the alleles in a gene. That is known as gene mapping, and it was given by Sturtevant. Okay, it was given by Sturtevant. His actual name is Alfred Sturtevant. So whenever this comes, that is gene mapping, just understand you're talking about Sturtevant. As easy as that. Now this is also a direct question. There's nothing up, you have nothing much to explain, you have to remember it. So what you can do is, children, you can make a list of people who are there. Okay, you take a page and you just write it, that this guy, this guy, this guy, this guy. If you want to remember in a fun way, write it in a fun way. Okay, just the points, nothing else, because in this chapter you need it. You know that, right? Okay, now next question, question number 6. The given diagram represents what? What is this thing showing? Oh, what is this round thingy? I don't understand, ma'am. Basically, this is the histone octamer that I just made. This is what? The histone octamer that I made. So basically you have 1, 2, 3, 4 histones at the front, 1, 2, 3, 4, total 8 histones, and on this histones you will get your DNA to be wrapping around. So your DNA is wrapping around twice, my dear children. So I am writing DNA wraps around twice. And one more thing, how many base pairs will be there in this particular DNA strand? What do you mean by base pairs? Basically, your DNA comes like this. So you have bases over here. A pairs with T, C with G. So how many pairs are there like this in this entire segment of rotation? Basically, 200 base pairs are there. This is also an important question. 200 base pairs of DNA will be there in one particular histone octamer. Okay. Now, this DNA will again go coil around with the next histone octamer that we are having over here. But before that, there is one more histone which is there, and you call this histone as H1 histone. What do you call this as? You call this as H1 histone, my dear children. This will wrap around this, and then it will go with the next one. So usually it is placed in distance. This is there, and between this guy is there. But when the DNA has to be made very, very short, it has to be condensed really, really well. At this point of time, everything of them comes together, and this is what is exactly shown over here. So what do you call this structure now? Where you have your histone proteins, where you have your DNA being wrapped around it. What do you call this structure, children? You call this structure as nucleosome. You call this structure as nucleosome. So this is one nucleosome, this is another nucleosome. In between the two nucleosomes, you will have this, that is in the H1 histone protein. So this becomes the correct answer. Okay, remember one more thing, that is, when, as I said, this association is kind of loose. Okay, this is here, that is there, and the middle one is there. So you call this as chromatin. Okay, where it just looks like beads on string like structure. On a string there will be small beads like this that will be visible, okay, under the microscope. Now when the chromatin condenses, okay, it forms something that is known as chromosomes. When it condenses, so when it comes close together, it is going to give a structure which is a rod like structure that you see, and it is known as chromosomes. Works? So what is a less condensed form? Chromatin. What is a more condensed form? Chromosomes. This is question number 6. Next question. Question number 7. A series of experiments were conducted by Frederick Griffith in 1928 on transforming principle with what? So an experiment was done by Griffith. Okay, Frederick Griffith, I told you that was the first guy who said that I want to find what exactly is the genetic material, my dear children. So he went on trying to understand this with the help of Streptococcus pneumoniae. with the help of Streptococcus pneumoniae. Now this particular bacteria, this is a bacteria, my dear children, and it has two strains. One is the R strain and one is the S strain. Okay, R strain is the rough strain, that means on the surface it has some rough thingy. S strain is the smooth strain which has an extra layer, polysaccharide layer around it, which makes it appear so smooth. So we think that this guy is the actual good guy. Okay, we think it's the good guy, but it's going to cause the whole trouble. Now what he did was, he took a mice, okay, and then first he put R strain of Streptococcus pneumoniae, which is usually going to cause pneumonia. But this guy, this particular mice was alive. So that means I can say that R strain is something that is not going to cause any trouble. It is non-virulent. It is what? Non-virulent. Then what I did is that I put the S strain. When I put the S strain, this particular mice, it died. It died, my dear children. So that means this S strain is the one that causes pneumonia. This one is not the non-viral. It is just a non-virulent one. It does not cause any trouble. This is the guy which is virulent. Okay, so he did that. You might be thinking why he did that. I'll tell you. Then afterwards, he did one more thing. He heat killed the S strain. Heat killed as in he treated this S strain with heat. Okay, now he put the heat killed S strain in the mouse, and then the thing that he found was the mouse was alive. He is not doing the same experiment. He is not doing the experiment with the same mice. Okay, you have 4 different situations. Okay, I'm too lazy to draw 4 different mice, that's why I'm showing it over here. So heat killed S strain was taken, and we assume since S strain is virulent, this particular mice should have died, but it didn't die because we heat killed it. So this didn't die, it was alive. So he was like, huh, what is exactly happening? I'm not able to understand. So then he did one more thing. He took heat killed S strain and the normal R strain, which was not causing any trouble. Now then he put it, and then he found that this particular mice is now dying. So he was like, I put the R strain, I agree, but R strain was non-virulent one, and I mixed it with heat killed S strain, which was also not causing any trouble before. And how did things happen? So probably it came to a conclusion that this heat killed S strain might be passing something to this R strain when they are mixed together, which is making this R strain convert into a S strain. And they were in a quest to find what was this thing that was being passed on. Even after passing them through heat, what was that thing that was able to survive or to be stable entirely during the whole process? Okay, later it was known that basically it was what? DNA. Because DNA is stable. DNA is a stable structure. So it survived and it got passed on to this particular thing, which made it an S strain, and now S strain was the reason why it was dying. So this was transformation principle. It was transforming the R strain to S strain, and this is what you call as transforming principle experiment. Okay, and it was done with the help of Streptococcus pneumoniae, my dear children. This came in 2022. This is the question number 7. Okay, so I hope you were able to understand. Now if you are not able to understand, since with the limited time, I won't be able to explain you too much, or else this whole lecture would become too long and you might not feel that urge to go through entire thing. So I have made a series for NEET children, okay, where I have explained it very much nicely for them. You can just watch it. Okay, you are also children, you're also studying from your NCERT, so it will be useful for you also. You can go and look for it. There is a playlist being made, there you will find it. Works? Now next thing, question number 8. The number of codons effective in coding 20 amino acids. Number of codons. Okay, you have to code for 20 amino acids, and codons basically means, for example, I have AUG. This is a codon, and codons, they are triplet, my dear children. So they have 3 nucleotides that come together to make a codon. Okay, so how many number of codons such like this should be there for coding 20 amino acids? That's the question. How many codons should be there as to code 20 amino acids? So the answer is 64. there are 64 codons that will code for 20 amino acids. So you said 264 and 20, I don't know, does not make sense, ma'am. So basically 64 codons are there where there will be multiple codons coding for 1 amino acids. There, for example, 1, 2, 3, 4 codon is coding for the same amino acid. Now next thing, 5, 6, 7, 8 is coding for the same amino acid, next amino acid. For example, this is coding for arginine, this will be coding for lysine, but 4 codons are being used for that, 4 codons are used for here. So basically 64 codons might look extra, but yes, they are going to code for 20 amino acids. Okay? Works. Now this is given in your NCERT. Now I want to tell you one very important thing, how we came to conclusion that it is 64 codons. Listen to me very, very carefully, okay? So basically, you know how many nucleotides are there? You have A, U, G, C. In the case of RNA, okay? In the case of DNA, you have A, T, G, C, okay? Now these are 4. So these 4 things, if you want to make it code for amino acids, that is basically 20 amino acids. So if I say 4 of them are coming in the pair of 2, I will be able to make only 16 amino acids. If they are coming in the pair of 2, I will be able to make only 16 amino acids, which is quite less. I have to make 20 amino acids. So now 4 of them, if they are making a triplet, so 4 is to the power 3, 64. Now it might be more than 20, but okay, as I said, the codons can code for the same amino acid. This is how we came to a conclusion. Okay, works? Cool. Now if they are asking you, you have to make 16 amino acids. And you have how many codons, my dear children? Suppose you have 2 nucleotides. You have 2 nucleotides, just A and U. And you have to make 16 codons. 16 amino acids. How can you make? So basically you say 2, if they come in the pair of 2, it will be just 4 amino acids. So this is cross. So 2, if it comes like this, it will be making just 8. 2, if it comes in the pair of 4, in the number of 4, if they are coming in 4 numbers, okay, then it will be, for example, AU, AU, it will be able to make 16, like that. But I don't want to go in detail like that, it might lead you to confusion. So as of now, you can just remember this is given in your NCERT as well. Cool. So 64 is the answer. We have 64 codons, including the stop codons and things like that. Okay. Question number 9. Which aspects form the basis of DNA fingerprinting? So DNA fingerprinting basically means that just like how we have different and unique fingerprints, similarly our DNA is also kind of unique. My DNA is kind of different from you because I have something different from you, and that something is known as satellite DNA. Rest whole of my DNA might be similar to you, but the satellite DNA, which is nothing but a non-coding DNA, this does not code for anything, this is the non-coding DNA, okay, which has so many repetitive units. They have so many repetitive, for example, ATGC. I have so many sequence of ATGC, 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 ATGC. Now, I am very unique with having this particular DNA. That is ATGC, ATGC, ATGC. Now, for example, I committed a crime and my hair follicles are there. Now, they did the testing and they found my DNA. They found the DNA and they found the sequence which was repeated again and again. They took that sequence. Okay, they took that sequence. ATGC was the sequence, okay, and now they made a complementary thing out of it, that is basically TACG. They made something like this. They made a probe like this. Okay, they know that this is the thing that is being repeated. They have made a probe like this, and now I, being a suspect, they will take my DNA out and try to check it. So when this probe, if it is going and attaching over here, and it is visible, that means probably I am the person who did the crime, because my DNA and the DNA that was found at the site of the crime is also similar. So this is a satellite DNA, and this forms the basis of DNA fingerprinting, my dear children. So satellite DNA showing high degree of repetition in DNA segments is the basis of DNA fingerprinting. You do not even have to go ahead with the next options. Okay, but the ratio of purines and pyrimidines in the DNA, no. The amount of DNA found in samples, no. The sequence of DNA present in the ridges and grooves of the fingerprints, no, my dear children. It's a satellite DNA. Just understand, DNA fingerprinting, it is for satellite DNA. Satellite DNA is what? Repetitive units, which is very unique in each of us. So you might have different repetitive unit, I might have different repetitive unit. Okay, got it? Okay. Next question. Question number 10. DNA replicates semi-conservatively was first shown in. So it was first shown in Escherichia coli. It was first shown in Escherichia coli, and I will tell you the name of the experiment also. This was Michelson-Stahl experiment. What is it, children? Michelson-Stahl experiment was done, according to which it was proven that DNA is going to replicate semi-conservatively. So I have the questions regarding that, so you will get a basic idea of it as well later on. Works? Cool? Now, going ahead with the next question. Okay, so this is the experiment. Should I show it now itself? Yes, I'll show it now. Anyways, you might be able to solve the answers later on if I tell you now itself. So basically what I did was, there is a type of nitrogen, okay, that is N15. What is it, children? N15. So I had a medium where I was growing the DNA and everything. So you know that nitrogen is a part of DNA. In DNA also you find nitrogen. So this particular N15 nitrogen got incorporated, and now I have a DNA strand which is N15 DNA. Now I will be having a DNA strand which is N15 DNA. If you didn't understand, I'll repeat it once more. So I had an Escherichia coli. I grew it in a medium which was rich in N15 isotope of nitrogen. Now the DNA which was there in this particular Escherichia coli had N15 in it as well. So that is the red color one. So this thing that you see over here, this is a N15 DNA. Now when I try to centrifuge it, okay, when I try to segregate it, okay, in what's this, cesium chloride medium, CSCl, yes. So it sediments down, okay, the DNA sediments down and comes out that it is a heavier isotope. So basically when you have two strands of N15, it will sediment somewhere here down because it has higher density. It has higher density. Cool. Now what I did was, I changed the medium. I used the medium which was rich in another isotope of nitrogen, that is N14 isotope. N14. So this time when the Escherichia coli is going to replicate its DNA, it is going to take use of the N14 nitrogen. Okay, because as I said, for making DNA you need nitrogen. So it is going to take N14 from the medium. Okay, so it takes 20 minutes for replication. The replication cycle of Escherichia coli is 20 minutes. So after 20 minutes when I saw, what did I find, children? I found that yes, the DNA has got replicated. The DNA definitely has got replicated. For replication, what do you need? This particular strand has to be separate, another strand has to be separate, and then you get a new strand. But this new strand did not have N15. Instead, it had N14 because you are growing in N14 medium. So now it was like this. One strand had N14, one strand had N15 isotope. This is shown over here. Now when I tried to centrifuge it in a cesium chloride medium, what I found was, in the test tube, it was settling somewhere here. It was not going down. It is not going down because both the strands does not have N15, just like it had. One had N14 and one had N15, which is why it was somewhere in the middle region. Okay, so I'll write N14 and N15. So I got to understand that DNA is replicating semi-conservatively as of now itself, because if it was not semi-conservatively replicating, then the DNA strands which were forming now would also have N15, but it is having just one parental strand. Okay, here also there is only one parental strand, so which is why I say it's semi-conservative. It's utilizing the parental strand and now it have a different strand as well. So it is semi-conservative mode of replication. But he wanted to go ahead. The Michelson-Stahl experiment, it went ahead. Okay. Now you again grew it in a media which has N14. Again you were allowing it to replicate once more. Then again 20 minutes. That means basically 40 minutes. 20 and then 20, that means basically after 40 minutes, if you say from starting, if you look at here, what happens is that basically this strand will segregate. So I have one strand like this, okay, one strand like this, and then I have one strand like this from here, and I have one strand like this from here. Now they are grown in N14 only. So this is the strand that they are forming. So now if you see, two of them basically is just made up of N14, N14, N14. Okay, two of them basically it has one parental strand from the N15 as well. So in a sense that I am trying to tell you is that now if I try to segregate, I'll be finding that two of them will be in the middle because it has both N14 and N15, here also N14 and N15. Okay, then there will be two strands that has just N14 and N14. So this is the lightest band that they observe. So they found two bands over here. They found, children, two bands over here, and they didn't find this higher density band over here. That means N15 and N15 was altogether gone. You just had N14 and N15 and also N14 and N14. That was the thing that you found. So this is why we say that it is semi-conservative. Again, as I said, when this divides, you get one strand from here, you get another strand from here, then you get two new strands which are both N14 and N14. You get it? Now, whatever you do, how many replications you do, every time you will get two strands which had N14 and N15, and other strands will be the new ones. Okay, this was the proof given by Michelson and Stahl. Okay, they said that yes, DNA is replicating semi-conservatively, and they showed it with the help of Escherichia coli. This came in 2022. Okay, works? Next question. Match the scientist of column 1 with the contribution in column 2. Chalo, let's do. So Griffith, what did he do, children? Remember Streptococcus pneumoniae? So Griffith is with transforming principle. Jacob and Monod is with lac operon. Michelson and Stahl, I told you right now, DNA replicates semi-conservatively. Hershey and Chase, DNA is the genetic material. It's easy, no, now. So Griffith is with 3. A is with 3. Jacob and Monod is with lac operon. So B is with 1. Michelson, rest everything does not match, right? So this is the answer. C is with 4. C is with 4, and Hershey and Chase is with 2. This is the answer. This came in 2021. Easy question. Okay, next. In which of the tRNA molecule is amino acid binding site located? So they are asking basically it's not which of the tRNA molecule, it's where in the tRNA molecule does the amino acid bind. So what do you mean by tRNA molecule? First thing that you should be knowing. So I said that mRNA basically, for example, AUG is there. Okay, so I said mRNA is that one thing which will be acting as a strand that will help in the formation of proteins. How do they do that? They come and attach to the smaller fragment of ribosome. This is the organelle which you call as ribosome. It has two subunits. One is the smaller subunit, and then there is one more subunit which is known as larger subunit. So just imagine a sandwich, okay? In between that you have your mRNA strand. Now what happens that there will be a tRNA that will be coming along. This tRNA looks something like this. This is the 5' end and this is the 3' end. This is also an this is also a RNA, right? So it will also have 5' and 3' end. Now this loop that you see, it is known as anticodon loop. That means it will be, if this is the codon, my dear children, which is going to code for an amino acid. You know what? The codons, they code for amino acids. You learned it, right? And these amino acids, when they come together, they give you proteins. They give the proteins. So basically AUG is over here. So anticodon will be containing the opposite of it. It will be UAC. So the anticodon is coming. Suppose one tRNA with the anticodon is coming, and it found that, oh, here I found my AUG, I will go bind there. If it is of complementary nature, it will go bind there. Now, why is it binding? What's the use of it? The thing is that on the 3' end over here, it has an amino acid attached to it. In which end, children? On the 3' end. On the 3' end, there will be an amino acid attached to it. So similarly, each codon will be read, and there will be more and more amino acids, my dear children, that will be attaching and that will be forming a protein at the end. Okay, so at what position in the tRNA molecule is the amino acid binding site located? It is the 3' end. Please remember, it is the 3' end. And what do you call this process as? You call it as charging of tRNA, or you call it as amino acylation. It's very much important, okay? What do you call it? Amino acylation, or you call it as charging of tRNA. Works, and you need an enzyme amino acetylase. That is an enzyme that comes. We will talk about that. There's a question regarding that as well. So please remember these terms, and please remember it's the 3' end that has the amino acid binding site. Works. Going ahead. 13th question. Okay. E. coli fully labeled with N15 is allowed to grow in N14 medium. Remember, E. coli that has N15, here also N15, is allowed to grow in a medium that has N14. Okay, the 2 strands of DNA molecule of the first generation bacteria have what? So now the 2 strands which would be forming after one replication, what would be that? So I say during DNA replication the DNA segregates. So I have one strand like this, one strand like this. This is N15. This is also N15. Now it is grown in N14 media. So the new strand that will be forming will be having N14. Here also it will be having N14, my dear children. So now when I was putting this alone in the test tube, I found that basically it was settling somewhere here because the density was quite high. But over here when I put them, it will be found in the middle because it has different density gradient centrifugation. Okay, that is something different. So that is happening in cesium chloride. So over here, because of the different density that is they are having, if it was N15, 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 then they would also have been settled somewhere here. But it's not happening like that. They are settling somewhere here because they have both the strands. Here also they have both the strands. So there's a different density that is being formed. So E. coli fully labeled with 15N is allowed to grow in 14N medium. The 2 strands of DNA molecule of the first generation, this is the first generation. After 20 minutes, you will get them. Okay, have same density and resemble with the parent DNA. Do they resemble with the parent DNA and do they have the same density, my dear children? No. Same density, again wrong. Different density, okay, agreed. But do not resemble with the parent DNA. Yes, they do not resemble. This is how the parent DNA looked. Now they do not have resemblance. It is semi-conservative. Half of the DNA comes as a new thing, right? So this is exactly correct. Let's read the next one. Different density but resemble with the parent DNA. They do not resemble with the parent DNA, my dear children. Okay, you have a new strand. Do not think like semi-conservative is there, so they would be resembling. No, you're not talking about that resemblance. Okay, you're talking about the structural resemblance. It is not structurally resembling. So this becomes your answer. Now, if you didn't understand the other portion, you won't be understanding this. But if you understood that, it will be really, really easy for you to understand this as well. And I hope my lovely kids understood that. And if you didn't, please pause the video, try to understand and then only go forward. Do not make it a rush, my dear children. You are in here for a preparation of a very important examination. Do not panic. Okay, do not panic. It's all good. You are trying and that's more than enough. Yeah. Okay, next question. Question number 14. Experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in chromosome was performed on which plant? Now, this is something that has high resemblance to this question. Okay, now this is a direct question from your NCERT which does not have much explanation in your NCERT as well. So they have mentioned one thing, that is, what are we using, children? Radioactive thymidine you are using. That is one thing. And you are trying to detect it in the distribution of newly synthesized DNA, just like how we were detecting N14. Okay, there we were using density centrifugation for understanding that. Here we are using radioactivity. Now, it was used in which particular plant? So the answer to this question is Vishya faba. It is Vishya faba. Now, where in NCERT you will find this? You will find it over here. Okay, so in your NCERT, after the Michelson style experiment, okay, at the end of it, in the next page, they have given this particular thing. You can read it, okay, at the topmost region. So very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in chromosome was performed on Vishya faba or faba beans by Taylor and colleagues in 1958. Now these things are very, very important. In NCERT, you tend to just not read these things out, so that's why we say NCERT is important. Please read, please read, please read. So I hope you got it now, why we say so. Okay, hope you got it. Next thing, question number 15. If the sequence of nucleotides in a template strand of DNA is, template strand, remember, and you have a coding strand. If in the template strand 3' to 5', the sequence is A, T, G, C, T, T, C, C, G, A, T. Okay, write the sequence in the corresponding region of the transcribed mRNA. Now it is an mRNA that is going to transcribe. It will be in 5' to 3'. So 5' to 3'. Let's make A. It is an RNA. In DNA you had thymine. In RNA you have uracil. So A will be binding with uracil and G will be binding with cytosine. Guanine will be binding with cytosine, adenine will be binding with uracil in the case of RNA. In the case of DNA, A was binding with T and G was binding with C. So over here it will be, sorry, again I have writing it wrong. So A will be binding with what, children? U, and T will be binding with A, right, C, G, U, U, G, G, C, U, oh sorry, here it is not U, here it will be A, here it will be U, and then again A, and this will be 3'. Match it, UAC, GAA, UAC, GAA, and then GGC, UUA. This is the answer. This is the answer. Okay, so why is this wrong, my dear children? Because it is 3' to 5' direction, but mRNA, it will be formed in 5' to 3' direction. You have to look for details. So usually look for, if you go like this and looking for answers, it is also same, just like that, but the polarity mentioned here is different. So this is the answer, my dear children. Works? Okay, next thing, question number 16. The length of DNA helix in a typical nucleosome is, I told you nucleosome, in an octamer, the DNA base pairs, how many of them will be there? 200 base pairs. Okay, in a nucleosome they are asking, they are not asking about the entire DNA. So in a nucleosome it is 200 base pairs. This is a direct question. Question number 16. Next question. 17. Which of the following types of RNA carries amino acids towards ribosome during translation? I told you what is translation. This is translation, my dear children. Where is it? Okay, over here, what is this strand which is carrying the amino acid? What do you call this type of RNA as? You call this as tRNA. tRNA stands for what, children? tRNA stands for transfer RNA. mRNA stands for messenger RNA. Messenger RNA carries the message of what proteins have to be made. So these are the ones that is going to get translated. rRNA is ribosomal RNA. Ribosomal RNA helps in the formation of the organelle, which is ribosome. Ribosome is very much important. So to form it, you need rRNA. dsRNA means double-stranded RNA. Double-stranded RNA, which is not needed over here. Okay, transfer RNA is transfer RNA because it is transferring the amino acids. So this becomes the answer. Next question. In eukaryotes, the entire base sequence of a gene does not appear in mature RNA because. What do you mean by that? Basically, I told you there is a DNA and this DNA is going to form mRNA and this mRNA is going to form protein, right? Why is this DNA directly not forming the proteins? That is the question. The thing is, this DNA is present inside what, children? It is present inside the nucleus. Who is responsible for making the proteins? It is ribosome. Where are the ribosomes, children? Outside the nucleus. If I send DNA outside the nucleus, there could be certain problems with the DNA which I do not want. It's my genetic material. I want to keep it really, really safe. So what can I do? The information which is there, I can transform it in some other form. Right, so that form is mRNA. But the first thing that you get initially when transcription happens, it's not directly forming mRNA. You call this RNA which is being formed over here as hnRNA, heterogeneous RNA. What is it? Heterogeneous nuclear RNA. So over here, hnRNA will be having, as I said, it is the RNA strand which will be just coding over here. For example, suppose this is a code that is being formed. This strand that is being formed here is hnRNA. Now, suppose until here it is coding for a thing. Okay, it is coding for a thing. Again, from here it is coding for a thing. All of this thing and this thing, when it comes together, it forms a protein, protein number 1. Now, I told you there are sequences which are present in between the non-coding sequences, which you call as introns. So, when I want to make proteins out of this and this, what I should be doing is that I should have to delete or basically cut this portion off, which is the intron portion. Okay, so initially I have an hnRNA. Where is it? Initially I have hnRNA which has both the exons and introns. Later I cut the introns off, which is known as splicing. This process is known as splicing, and now I just have exons, the coding ones, and I call the sequence of exons as mRNA, that is messenger RNA, which is the mature form of RNA. So this is a mature form, we can say. This is the mature form that we want. So in eukaryotes, the entire base sequence of gene does not appear in the mature RNA. So entire sequence is not coming. This sequence is lost. Why, children? Because some gene sequences are removed by exonuclease. Some gene sequences are removed, I agree, but it is not by exonuclease. Now that means you should be knowing what is exonuclease as well. Exonucleases are those which would be cutting DNA at the ends. So here I am not cutting DNA at the ends, I am cutting from the middle region. So I need endonucleases. I need what? Endonucleases, not exonucleases. This is wrong. Transcription in eukaryotes consumes more energy, does not make any sense. Coding sequences are removed during the process. If you remove coding sequences, my dear children, who will make proteins? This is also wrong. Interons are removed during the processing, and this is known as splicing. This is the answer. If introns are removed, which is why you do not see all the base sequences in the mature RNA, my dear children. Works? Okay? Next. Question number 19. When Escherichia coli cells are cultured in a medium where lactose is absent, okay, it's absent. When Escherichia coli, E. coli, they are cultured in a medium where lactose is absent, my dear children. Lactose is not there, okay? I'm writing. Okay. The I gene of lac operon continues to produce repressor mRNA because it is what? What is I gene? What is this thing, ma'am? Basically, the lac operon, which was found by Francois Jacob and Monod. Okay, what was it all about? So, basically, there were three, gene sequences over here. This whole thing was a structural gene. This whole thing that you see is a structural gene. Over here, there is the promoter region of this particular structural gene. Okay, then there is another gene over here which is known as I gene, and here you have the promoter region of I gene. Okay, I gene basically it is going to inhibit the function of this particular gene. So these genes, when they show their function, they are going to code for enzymes, enzyme A, enzyme B and enzyme C, which are very much important in the breaking down of lactose, in the taking up of lactose and things like that. Okay, they need this particular Escherichia coli. This particular Escherichia coli is needing lactose as an energy source. Okay, it is needing lactose as an energy source. Now, what happens is that when lactose is available at this point of time, the first enzyme which is there, which is beta galactosidase, it will be breaking down the lactose. The second enzyme, which is permease, it is the guy which helps the E. coli to take in the lactose from the medium. So it makes permeable pores over here. So that is the use of permease. The third one is transacetylase, which would be dealing with everything that comes in between. For example, any metabolites which is not needed, it will be structuring out and things like that. So these enzymes are kind of really important, and this is the promoter sequence for this particular enzymes, okay, for this particular gene. When this particular gene has to get transcribed, that means this promoter should be active. But the gene next to it, that is I gene, it is producing some protein, okay. This is responsible for coding for a protein that is known as repressor protein. What is it, children? Repressor protein. And what does this repressor protein do? It goes and binds on the promoter. This guy goes and binds on the promoter such that transcription of these genes does not happen. If I do not get these genes, nothing will be taking place there. Nothing will be taking place there. So usually always this repressor protein is going and binding over there such that none of this is becoming active. But when I have put lactose in the media, what happens is that the permease which was produced initially, and I'll tell you one more thing, even though this guy is bound over there, there will be little amount of transcription still going on, very little, okay, very little, okay, which is why we have little amount of permease, and this permease allows lactose to get in. When lactose comes and sees that this is happening with the enzyme which is going to break me, what happens is that this lactose goes and binds with the repressor protein. It will be binding with the repressor protein such that this particular repressor protein would not be able to bind to the promoter, and now the whole reaction or basically transcription can take place, and you can get all these enzymes, and the lactose can be broken down to give energy to this particular thing. So this is the usual thing. So over here I say, what is the role of I gene? What is the role of I gene? Is this always forming repressor mRNA? It is going to form repressor protein which is going to bind on the promoter and does not allow the transcription of this thing, that is lac operon. So basically, what can I call it? Yes, it is a structural gene for sure, but in your NCERT they have mentioned something according to which I would call it a constitutive gene. A constitutive gene is one that is continuously producing its thing, that is a repressor protein, and then this is continuously working on your other structural genes as well. This is continuously doing its job, which is why I call it constitutive gene, my dear children. It's a structural gene. This is a structural gene, this is also a structural gene, but over here I call I gene as constitutive gene because this is continuously showing its function of repressing the properties over here. Okay, now if you do not believe me, this is something from your NCERT. So in NCERT, this is the picture that is given over here. Z, Y, A are the name of the genes. Z is going to code for beta galactosidase, Y is going for permease, and transacetylase is A. Okay, now over here I told you that is I gene is there. The repressor of the operon, listen, read it carefully. The repressor of the operon is synthesized all the time constitutively from the I gene. You get it? The repressor is formed constitutively all the time, which is why I call this as a constitutive gene, my dear children. Works? Okay, it's very important, you have to remember it. Now, 28th. For the given sequence of DNA, identify the complementary sequence of bases on its mRNA from the options below. Same question, I think. It's kind of same question. Okay, they have just changed the option and this came in separate year. So, this is the sequence that we have. We have to make a complementary strand of it. Then only we can make an mRNA. So, this has to be 5'. A is here, so it will be U here. T is there, so it will be A here. It will be C here. It will be G here. It will be U here, A here, C, G, again U, A, C, G, and this will be 3'. So 5 UAC, 5 UAC, GUA, CG, UACG. This is 5' to 5'. This becomes wrong. You get it? Okay. Now let's see, is there some other option or probably there's a typing mistake. For the given sequence of DNA, identify the complementary sequence of bases on its mRNA from the options below. Yes, this is the DNA, the complementary. So it should be 3'. This is 3'. Okay, there's a typo error over here. If it's 3', this becomes the answer. Okay, rest all are the wrong options. Okay, rest all are the wrong options that you're having. This is the answer, same like that. Okay, nothing different. Now 21st, which of the following amino acids is coded by a single codon? I told you there are multiple amino acids. There are multiple codons that could code for a single amino acid. Okay, now the question is, there is an amino acid that is coded by just a single codon. What is that? And the answer is tryptophan, my dear children. This is the only amino acid that is coded by a single codon. So over here, if you have seen this carefully, you see phenylalanine is coded by 2 codons, leucine is coded by 1, 2, 3, 4, 5, 6 codons, isoleucine is coded by 3 codons, methionine is coded by AUG. Okay, that is also one more thing that is coded by one particular codon. Okay, but in this option you are asked about to choose from them. Okay, so here you just have one guy, that is tryptophan, that is what I meant. Okay, so methionine is gotten by AUG, and then for all of them, if you see, for all of them you see there are multiple codons coding for the same amino acid, but tryptophan it's UGG, and that's the only amino acid like that. Okay, like methionine, I always tend to forget it, but yes. So the answer is tryptophan. If you can remember the codon, it will be really good. UGG. There's a fun fact about methionine, I'll tell you that. So AUG, which is a codon that codes for methionine, is the same codon that is known as start codon. So basically, when the tRNA comes with an anticodon loop and binds with it, when they see this particular codon, that's when they start the transcription, which is why it is known as start codon. So AUG codon has two functions. One is to code for methionine, the other is to act as a start codon. Okay, so these things about codons you have to remember. What is the name of the start codon? It is AUG. What is the stop codon? So whenever a tRNA sees a stop codon, it will stop forming the protein. Okay, you have UAG, UGA and UAA. Okay, these are the stop codons that you are having. So stop codons over here, UAA, UAG and UGA. UAA, UGA, UAG. Works? So these are the stop codons. Please, where is it, children? You have to remember it. Uga, uag, ua. Okay, uga, ua, uag. Fine? And methionine and tryptophan are the only ones which has just one amino acid, one codon coding for them. Cool. Let's go ahead. You will be finding questions regarding this. Okay. In prokaryotes, I'll recommend you to learn this. Very important. So in the NEET video, I have told you a way that you can follow to remember this. You can watch that particular video if you want. Now, in prokaryotes, the transcription of DNA is initiated with the help of. In prokaryotes, the transcription of DNA. So this is the DNA, okay, and I say this is a 3', 5', this is 5', 3', this is the template strand, this is the coding strand. So over here there is a sequence which is known as promoter sequence, my dear children, and over here there will be a sequence that is known as terminator sequence. So on this promoter sequence, the RNA polymerase has to come and bind. But it will only bind if there is a very important factor being present along with it, which is known as sigma factor. So sigma factor is needed for initiating. Sigma factor is also known as initiator factor. Okay, so it is needed for initiating the process of transcription. Over here you have another factor which is known as rho factor, which binds over here and terminates it. This is terminator factor, which is rho. So answer is sigma. For initiating you need sigma. Next thing, 23rd question. According to Human Genome Project, the total number of genes in human genome is estimated at 30,000. The number of genes on Y chromosomes are. So the Y chromosomes has least number of genes, my dear children, and the chromosome number 1 has highest number of genes. So chromosome 1 has highest and Y gene has the lowest. Your homework is to find how many numbers of genes are there on chromosome number 1. I'll tell you about Y gene, that's the question. It is 231. Okay, this came in 2019. This is given under Human Genome Project. Works? Next question. In a crime investigation, the investigating officer collects different biological samples from the crime spot. Okay, they took different samples from the crime spot for DNA fingerprinting analysis. Which of the following samples is not helpful in this analysis? Which one is not helpful in this analysis, my dear children? So he took skin shreds. Is it okay? Is it okay? Can it help in finding the culprit? Yes, because in it has nucleus, it has DNA, so it's fine. Semen sample, yes, it also has DNA, it will be helpful. Hair follicle, definitely yes. Erythrocytes, a big no. Erythrocytes is basically RBC, my dear children. And RBC, you know what? Since their only function, major function is to transport oxygen, they have hemoglobin. So to accommodate more and more hemoglobin, they said we do not want nucleus. So they are the cells which do not have nucleus. If no nucleus is there, can I accommodate DNA? So there is no DNA. If there is no DNA, there is no DNA fingerprinting as well. So this becomes the answer. Works? Next question. A mature mRNA consists of 900 bases without any stop codon in between. Calculate the number of amino acids coded by mRNA during translation. So 900 bases are there, my dear children. 900 bases are there, and you have to find the number of amino acids. Number of bases is 900. Number of amino acid will be what? So you know bases comes in triplets. Okay, so 900 divided by 3, they will be coming in 3-3 format. Okay, so if I found that, I get 300. So there will be 300 codons. There will be 300 codons. Okay, there will be 300 codons, and these 300 codons will be coding for 300 amino acids. I'll explain once more. You have 900 bases. These bases comes in the pair of triplets. So not in a pair of triplets, they come in the form of 3 groups. Okay, so 900 divided by 3, I get 300 of them. So 300 codons are there. For example, AUG is 1, AUG is 2, that is 1 codon, this is next codon. So similarly, you have 300 of them. So each codon basically is coding for just 1 amino acid, right? So I have 300 amino acids. So this becomes the answer. Works? Next question. DNA present in the nucleus was named as nuclein by whom? Who was the person who said that it was nuclein? It was Frederick Mischer, my dear children. Okay, it was Frederick Mischer who was the person who said that it was nuclein. He was the first guy to initiate a lot of things. This is a direct question from NCERT. You can remember as such. Works? 27th. When does lac operon in E. coli become switched on? So it is usually switched off because the repressor protein is always there. When does it become switched on, my dear children? When the lactose comes in. When the lactose comes in, it binds with the repressor protein, and now the repressor protein would not be able to bind to the promoter region, and hence transcription would be taking place there. So when lactose is there, that's when it becomes active, or when it gets switched on. Usually it is switched off. So repressor protein binds to operator? No, children. When it binds to operator region, that's not when things work. Okay, I missed one thing. It's not the promoter region, it's the operator region. Okay, where is it? There is one more region which is the operator region where it goes and binds. I missed this thing. That's what I was thinking. So this is the promoter region where the RNA polymerase comes and bind. Then there's an operator region where this guy comes and bind, the repressor protein. Okay, please remember operator. Promoter region, RNA polymerase comes and bind, but it won't be able to move ahead because on the operator you have the repressor protein. PO. Okay. Thank you, question, for making me remind this. Now, RNA polymerase binds to operator. No, my dear children, lactose is present and binds to the repressor. This is present. Okay, lactose is present, it binds to the RNA polymerase. No, children. This is the correct answer. Works? Next question. Match the process of column 1 with enzyme involved in column 2. So, DNA replication needs what? DNA polymerase. Okay. Transcription, it involves RNA polymerase because you are making RNA out of a DNA, right? Translation, you need an enzyme, I told you, amino acyl synthetase for amino acylation. Translation is a process where the tRNA brings in amino acid. For this, it needs amino acyl synthetase. for amino acylation, that is for carrying this AA, that is amino acid, and then only the whole process of translation will be happening. Reverse transcription is a process where usually what happens, you have DNA. This DNA helps in the formation of RNA. In the case of reverse transcription, RNA is going to form DNA. So that is kind of different. So DNA replication, so I will say, so for this you need an enzyme that is known as reverse transcriptase. So reverse transcription, you need reverse transcriptase that is used in HIV. You might be knowing about that, right? Now, so A with Q, everyone is with Q only. Translation with S, so Q with S. So I have these two options. I can cut these because it is not S. Next, C is with P. C is with P. I just have one option like that, and hence it becomes the answer. Easy, no? Yes. Next question. The codon on mRNA are CAU, CCU, AA, AAA, CUG. Now you had to tell the answer. How will you tell the answer if you're not aware about the codons? So we have to remember the codons. CAU stands for histidine. CAU stands for histidine. So let's see. Okay, just a second, okay. CAU, CCU, okay, CAU, CCU, AAA, AAA, okay, wait. Where is it? CAU, as I said, it is for histidine. Okay, CCU, it was asked, is for proline. And then AAA is there. Where is AAA, my dear children? It will be somewhere here. Yes, is for lysine. So with this, what do you get as answer? Okay, okay. CAU is for histidine. CCU is for proline. AAA is for lysine. Okay, so with this one only you can get the answer. Histidine, proline, lysine should be there. Histidine, proline, lysine, and then it will be leucine. Let's see, CUG is for leucine or not? CUG is for leucine. Okay, so this becomes the answer. Now if you're not aware about this, you won't be able to answer this. That's why I told you can watch that video. I've given a short trick to remember, okay, which I forgot now, otherwise I would have told you now itself. So you can use it. Now, the anticodon found on the tRNA for tryptophan amino acid is, remember tryptophan, my dear children? Okay, remember tryptophan? What was the code? UGG. So UGG is the code for tryptophan. So in the mRNA there should be UGG. What is there on the anticodon loop? That is the question. So in the anticodon loop it should be A, C, C. A, C, C, my dear children. So I will write it properly because you will be getting the PDF of this. So A, C, C. Works? So this becomes the answer. You're talking about anticodon loop. Remember, the codons are not the ones that is present on the anticodon loop. Anticodon will be having anticodon. The codons are present on the mRNA, and in the mRNA, if it is UGG, children, here it will be this. Okay? So I hope you understood all the questions, my dear children. You can practice a lot more. Okay, you can, it's not like you just have to stick on to this. You can watch other videos as well for NEET also if you want to crack, because anyways you are trying to understand the basic concepts of your NCERT. You can take the help of that. Okay, so that's all. And if you think that this particular video was very helpful for you, definitely do not forget to subscribe to our channel, that is Diksha Karnataka, and also put down a sweet comment. I would love reading it. Yeah, and I wish you all the very best. Do not be panicked. Everything is going to be really, really good. Just trust your instincts and give your best. So bye bye.